Today we'll talk about the fluid mosaic model, what it is, and why it's important. We will not be covering the different forms of active and passive transport, because that's a topic for Unit 1. This course just focuses on Unit 3 and 4. This video covers this dot on the study design. When we talk about the fluid mosaic model, we're talking about a membrane of a cell. Membranes surround all cells, like this little guy, and create a barrier between the inside and the outside of the cell. The membrane is so important for cellular functions because they allow the cell to respond to their environment in many ways, such as evading or fending off danger or growing. Let's take a look into the structure of a membrane a little bit more closely. The phospholipid bilayer is the basic structure of a membrane, and it is comprised of phospholipids. These are described in more detail in our biomacromolecules video, so hop on over there if you need any more help with this. These molecules bind together because they're all hydrophobic. They fear water, and this allows for the other elements to be integrated into the membrane. They are much like bricks in a house. You can't have a window without a wall to put it in. The plasma membrane is described as fluid because it is not a rigid structure that holds a single form. It moves and bends, kind of like rubber. The membrane is referred to as a mosaic because it's made up of lots of different components, much like a mosaic artwork. The components can switch places and move within the membrane, which gives it this fluidity. Let's put this together in context and outline the functions of each of these components. Firstly, we have the phospholipid bilayer, and as we discussed, this forms the basis of the membrane. Most of our other components are integrated into it. Integral proteins are proteins that span through the membrane and cannot be removed. Try to think of these as permanently integrated into the membrane, and if you remove them, you would break the membrane entirely. You can have many different kinds of integral proteins, such as membrane spanning, which means they go all the way through the membrane, inside to out. And these can be channels, which can allow substances to move into or out of the cell. You can also have other kinds of proteins, which are embedded in the membrane, but only on one side. They don't span the whole way through. You can have ones that are on the outside or on the inside. Glycoproteins and glycolipids. These look like little strings of beads, and they protrude out from the membrane. They are important for cell-cell communication. Really, the only thing you need to think about in terms of what's different between these two is that glycolipids are these glyco groups that sit on the heads of phospholipids, and glycoproteins sit on top of proteins embedded in the membrane. These are really important for immunological communication. Peripheral proteins can also come in and bind to the membrane or other membrane proteins, but they are not permanently integrated. They can come and go as they please. Hormones are a good example of these, as they can bind when needed and disassociate when they're no longer required. Cholesterol is an important structure in the membrane, and it helps to regulate the fluidity of the membrane. The more of it that we have, the more rigid the membrane becomes. Too much of it is bad, as the cells get too stiff, but too little is also not great, as the membrane can start to become too loose and lose its integrity and strength. We can also find receptors in the cell membrane, and these are a kind of integral membrane protein that binds specific ligands to send a message inside the cell from its external environment. We'll touch on this a little bit more depth in a later video when we talk about signal transduction. G proteins are our last and final component that we'll discuss here, and these reside inside the cell. They help to transmit the messages from the external stimuli by assisting with the amplification of a signal. But this again will go into more detail on our signal transduction lesson. Thanks for watching this week's video, and don't forget to give us a thumbs up and subscribe for more free biology tutoring videos. Head over to our website for a free downloadable PDF of the notes for this lesson. Still need more help? Contact us here and book a session with a tutor. We do Skype, private and small group classes. Use the code YouTube for your first free session. See you in the next one.